Because frugivores are a major danger to seeds, it is necessary to think about how edible fruits evolved. They must offer some advantage that outweighs the danger. Animals have predictable habits and migration patterns. Ants, birds, and mammals carefully select the sites where they feed, nest, and sleep. Therefore, seeds are not distributed at random but are moved through the environment to specific sites. One example is mistletoe. Its seeds are sticky and adhere to a bird's beak as it eats the fruit. The bird probably feeds and cleans itself in the same species of tree, thus, the seeds are rubbed off the beak directly onto a proper host tree. Few seeds are ever lost by dropping to the ground or being deposited in the wrong tree, as would happen with wind or water dispersal. Just as efficient is dispersal of fruits and seeds of marsh plants. The birds and animals that live in marshes typically spend little or no time in other habitats, so seeds carried by these animals are almost certainly distributed to favorable sites. Figure 9.33 At maturity, different layers in this dry fruit contract in opposite directions, causing the pod to twist and break open. As these maple fruits drop, the wings cause them to rotate and float, thus remaining aloft and traveling much farther on the wind. The spiral, hairy tails of these geranium fruits let them float on the wind over long distances. A special benefit of seed distribution by animals is that the deposited seed may find itself in a small mound of organic fertilizer. Seeds adapted to pass through an animal's digestive tract are resistant to digestive enzymes and can tolerate the high level of ammonium in fecal matter. Because many seeds of plants cannot tolerate these two factors, the adapted seeds find themselves in a microenvironment that is not only nutrient-rich but also excludes some competitors. Figure 9.34 This seedling is as happy as a clam at high tide. It has enough fertilizer to last for months. Many of its competitors have been digested, others are being suffocated. At the next level. 1. Pollination syndromes. Each type of pollination agent affects the evolution of the flowers it pollinates. Wind-pollinated flowers usually evolve to lack sepals and petals. Hummingbirds prefer flowers that are long, tubular, and red. Bats fly at night, and bat-pollinated flowers evolve to open after sunset, to have a strong fragrance, and even to reflect the bat's sonar back to the bat such that the flower is found easily in the dark. Dozens of other pollination syndromes occur and are remarkable examples of coevolution between plants and animals. Be sure to search for pseudocopulation between wasps and orchids. 2. Frugivores and seed dispersal. Coevolution has occurred between plants and animals such that seeds are dispersed to preferred sites rather than at random. For example, seeds of epiphytic plants and parasitic plants should be carried to new branches of suitable host trees, usually by having birds eat the fruits without damaging the seeds. As with pollination, many syndromes have evolved, some involving fruit as a reward for dispersal, others involving food bodies on the seeds themselves. Of special interest, several plant species produce edible fruits, but we have not found any animals that eat them. One hypothesis is that the animals went extinct recently, but the plants persist even though their seeds are no longer being dispersed effectively. Be sure to search on extinct seed dispersers. 3. Evolution of flowers. In most flowers, both stamens and carpels occur together within the same flower. Perfect flowers are common, and evidence indicates that the earliest flowers were perfect. But in all other seed plants, pollen cones and seed cones are always separate. Pollen-producing structures never occur together with ovule-producing structures. Thus, we are still far from being certain of the steps involved in the evolution of early flowers, the early angiosperms, and their ancestors, but this is a fascinating area. There are conflicting theories and conflicting evidence. This is a challenging topic for anyone, especially for beginning botany students, but it is very rewarding. 4. Recommended books. A team of botanists and a visual artist created 3 wonderful books about plant reproduction. These are large coffee table books with large illustrations, each image being a photograph or a scanning electron micrograph of pollen or fruits or seeds. SEM images are naturally black and white, but the team colorized the images to enhance certain features so that the various parts are more readily understandable. Each is accompanied by just enough text to fully explain the biology without overwhelming the reader. I recommend them highly. Pollen, The Hidden Sexuality of Flowers by Rob Kessler and Madeline Harley. Fruit, Edible, Inedible, Incredible by Wolfgang Stuppy and Rob Kessler. Seeds, Time Capsules of Life by Rob Kessler and Wolfgang Stuppy. Summary. 1. Asexual reproduction results in new individuals genetically identical to the parent. The parent and all progeny are equally adapted to the habitat. 2. Sexual reproduction results in progeny that differ from each other genetically. There is a range of fitness, and some progeny may have a combination of maternal and paternal genes that causes them to be even better adapted than the parents. 3. All seed plants have a life cycle that consists of an alternation of heteromorphic generations. Microgametes and megagametes are produced by microgametophytes and megagametophytes that grow from microspores and megaspores. Most species have only one type of sporophyte, capable of producing both types of spores. Dioecious species have two types of sporophytes, one of which produces microspores and the other megaspores. 4. Flowers contain all or some of the following non-essential organs: pedicel, receptacle, sepals, and petals, and essential organs, stamens and carpels. 5. In flowering plants, the microgametophyte consists initially of a vegetative cell and a generative cell, but the latter divides into two sperm cells. The megagametophyte consists of the egg cell, two synergid cells, a binucleate central cell, and three antipodal cells. 6. After fertilization, the zygote grows into an embryo. Polar nuclei fuse with the second sperm nucleus, establishing the endosperm nucleus and endosperm, and the integuments develop into a seed coat. The entire structure is a seed. The ovary develops into a fruit. 7. Inflorescences are groups of many flowers. They may be determinate or indeterminate and may contain several types of specialized flowers. 8. Fruits protect seeds during development and may aid in their dispersal and maturation. Fruits are either dry, inedible, dehiscent, or indehiscent, or they are fleshy, edible, and indehiscent. Important terms. Alternation of generations, anther carpels, coevolution, compatibility barriers. Complete flowers, cotyledons, cross-pollination, double fertilization, embryo, sac, endosperm, epicotyl, fruit, gametes, gametophyte. Heteromorphic generations, hypocotyl, imperfect flowers, incomplete flowers, inflorescence, karyogamy, ovary, ovules, pedicel, perfect flower, petals, plasmogamy, pollen, pollen tube, radical, receptacles, seed coat, seed sepals, spores, sporophytes, stamen, stigma, style, syngamy, zygote. Review questions. 1. Reproduction can serve two very different functions. What are they? 2. Under what conditions is it selectively advantageous for a plant to produce offspring identical to itself? 3. Imagine a plant that has been well adapted to a particular habitat, and it has been reproducing only asexually, thus, all of its offspring are identical to it. If the climate or other conditions change such that the plant can no longer survive in the new conditions, what will happen to all of its offspring? Why? 4. With asexual reproduction, are progeny ever more fit, more adapted than the parent? 5. Look at the apples in Figure 9, 1a, all growing on a single tree. Do all of the seeds in all of the apples in the photograph have the same maternal parent? Do they all have the same paternal parent? 6. Sexual reproduction produces offspring that are not identical to each other or to either parent. Usually, some are more well adapted than the parents. Some are more poorly adapted, and most are about as adapted. What is one of the beneficial aspects of this diversity? 7. In stable populations, ones that are neither increasing nor decreasing in abundance. About how many of a plant's seeds survive and grow to adulthood, being able to replace it when it dies? If during the plant's lifetime it produces 100,000 seeds, how many do not survive, do not grow, and cannot replace it when it dies? 8. Describe the life cycle of us humans. Are the tissues and organs in our bodies made up of diploid cells or haploid ones? Our reproductive organs make sperm cells in males and egg cells in females. Is this done by mitosis or meiosis? When our bodies make gametes, does every cell in our body become a sperm cell or an egg cell, or do just some of the cells in our reproductive organs do this? Can our haploid sperm and egg cells undergo mitosis and grow into new animals that look like us but are haploid instead? After a fertilized egg is formed, is it diploid or haploid? Can it immediately undergo meiosis to make 4 new sperm cells or egg cells, or can it only grow by mitosis into another person? 9. The plants we are familiar with are called sporophytes or the sporophyte generation. Do these plants have bodies made up of diploid cells or haploid ones? In which organs does meiosis occur, leaves, stems, stamens, roots, carpels, or petals? When plants undergo meiosis, do all cells of the plant undergo meiosis? Do all cells become haploid, or does meiosis occur in just a few cells and some flower parts? 10. In animals, meiosis produces gametes, but that does not happen in plants. When some of the cells of a sporophyte undergo meiosis, what types of cells are produced? What do they grow into? 11. What is a gametophyte? How many different types of gametophytes are there in a plant life cycle? What do they look like? Do the sporophytes and gametophytes of seed plants ever look like each other? 12. Draw and label a microgametophyte of a flowering plant. What type of gamete does it produce? Draw and label a megagametophyte of a flowering plant. What type of gamete does it produce? 13. Flowers typically have many parts, although some flowers can be missing some of the standard parts. What is the name of each of the following parts? A. The stalk of the flower. B. The end of the stalk, where the other parts are attached. C. The parts that are usually green and protect the flower bud as it develops. D. The parts that attract pollinators. E. The parts that produce pollen. F. The parts that receive pollen and which contain ovules. 14. If a flower has all of the parts C, D, E, and F in question 13, they are said to be flowers. If they are missing any of these parts, they are said to be flowers. Parts E and F are especially important because they produce the reproductive cells, the spores. If a flower has both E and F in question 13, they are flowers. If they are missing either E or F or both, they are flowers. 15. In flowers that are pollinated by wind or water, which of the parts in question 13 is often missing? 16. What is the difference between a perfect and an imperfect flower? What is the difference between a complete and an incomplete flower? 17. A stamen usually has 2 parts. The stalk is called A and an upper portion, the, which produces the pollen. Only some of the cells in the upper part undergo meiosis and become pollen grains. Those cells are called mother cells or neighboring anther cells, in a layer called the act as nurse cells. 18. Carpels usually have 3 parts, a that catches pollen grains, A. That elevates the first part, and in where megaspores are produced. In this last part, there are placentae that bear small structures called, each with a short stalk called a funiculus and a central mass of parenchyma called the nucellus. One cell in the nucellus will be the mother cell or 19. The megaspore in most flowering plants grows into a megagametophyte that has 7 cells and 8 nuclei. Name and describe the 7 cells. 20. After pollen lands on a stigma, it is far away from the ovule with the megagametophyte, which holds the egg. How are the 2 sperm cells transported from the stigma to the egg? 21. In angiosperms, as a sperm cell enters the egg, it loses both and such that only the sperm cell nucleus contributes any DNA to the new zygote. In gymnosperms, however, the sperm cell loses only its such that the zygote inherits both plastids and a nucleus from the sperm cell. 22. After the sperm cells enter a synergid, one fertilizes the egg in a 2-step process. First there is fusion of the sperm cell's protoplasm with that of the egg, a step called. Then the sperm cell nucleus fuses with the egg cell nucleus, the second step, called. 23. What happens to the second sperm nucleus, the one that does not fertilize the egg cell? What is the tissue that develops from this second fertilization? How is coconut related to this? 24. In most eudicot seeds, the parts of the embryo are very easy to see. Describe each of these parts: A. Cotyledons, B. Radicals, C. Hypocotyl, D. Epicotyl. E. Extra credit portion. Answer the following question if you have read about the radicle with regard to a taproot system and a fibrous root system. How is the radicle involved in these different types of root systems? 25. What are albuminous and exalbuminous seeds? Consider corn, peas, and beans. Which of these seeds are which? 26. After pollination and fertilization, as the ovule develops into a seed, the ovary matures into a. Many of these have 3 parts. The is the skin or peel. The is the flesh, and the innermost layer, the may be tough like the pit of a cherry. 27. What is the difference between self-pollination and cross-pollination? If pollen is transferred from the stamens of a flower to the stigma of the very same flower, is that cross-pollination or self-pollination? If it is transferred from the stamen of one flower to the stigma of another flower on the very same plant? If it is transferred from the stamen of one flower to the stigma of another flower on a different plant, but a plant that is a clone of the first one? Animals do not have pollen, of course. But why is it that most animals never have to worry about the equivalent problem of self-fertilization? Why cannot most animals fertilize themselves? 28. Describe a species that is dioecious. Name several examples. In order for sexual reproduction to occur in a dioecious species, how many separate plants must be involved? Name them all. 29. Describe a monoecious species. Name several examples. 30. In several pairs of plant and animal species, plants have become modified such that only its animal partner can pollinate it, and the animals have become modified such that they are especially well adapted to pollinate just their plant partner. What is the name of this type of evolution that results in 2 organisms becoming particularly adapted to and dependent on each other? 31. Explain the following terms: inferior ovary, superior ovary, actinomorphic flower, zygomorphic flower. How is each of these modifications selectively advantageous? 32. What is an inflorescence? The inflorescences of a sausage tree hang far down out of the tree. How is this of benefit to bats? Why can the inflorescence of Combretum attract more pollinators than can the individual flowers? 33. After pollination and then fertilization, what usually happens to each of the following: stigma, style, carpel, ovule, integuments, and zygote? 34. Fruits are often classified as dry or fleshy. What is the difference? Which of these 2 are dehiscent, which are indehiscent? 35. In ordinary English, we use the word fruit to mean something sweet and juicy. However, the following things are fruits: peanut shells, pea pods, bell peppers, and chili peppers. What is the characteristic that lets us know these really are fruits even though they are not sweet? In contrast, bananas are fruits that do not have this characteristic. 36. Some things that we call fruits are not true fruits but instead are accessory fruits. What is the red part of a strawberry, and what are the true strawberry fruits? In an apple, what is the fleshy part that we eat, and what is the core that we throw away? Design credits: Hummingbird, Tongo 58 Moment Getty, Green Plant Cells, Shutterstock Incorporated Natalia Ora, Purple Tulip, Shutterstock Incorporated Marie C. Fields, Dandelion, Shutterstock Incorporated Daniel Kreisel, Poppy, Shutterstock Incorporated Saryuri, Plant Icon, Shutterstock Incorporated Vector, Digging Man Icon, Shutterstock Incorporated Z-Art. Part 2. Plant Physiology and Development. Chapter 10 Energy Metabolism, Photosynthesis. Chapter 11 Energy Metabolism, Respiration. Chapter 12 Transport Processes. Chapter 13 Soils and Mineral Nutrition. Chapter 14 Development and Morphogenesis. Chapter 15 Genes and the Genetic Basis of Metabolism and Development. Part Opener Image: Plants often develop in wonderfully diverse ways. The main branch here grows horizontally, but all its branches grow upward. Notice too that only axillary buds on the upper surface of the horizontal branch have grown out, whereas all those on the sides and lower surface are still dormant. All axillary buds in the vertical branches are dormant. As these branches grow, they perceive and respond to conditions around themselves. Almost certainly the important environmental cue is either sunlight or gravity. Can you design an experiment to determine which factor is controlling the physiology of these branches? Araucaria subulata. When you are studying plant structure, you can often see the material directly with the naked eye, although light or electron microscopy may be necessary to see some structures. In plant physiology and development, however, the objects of our study, chemical reactions and metabolic pathways, cannot be seen at all. Instead, the results of experiments, measurements, and analyses are studied, and hypothetical reactions and pathways are set down on paper. From these, predictions are made and new observations planned to test the hypotheses. It is easy for us to study and memorize metabolic pathways, chemical formulas, and diagrams of physiological control mechanisms without appreciating that these have never been, and cannot ever be, seen directly. Every one is a theoretical model that is consistent with the majority of the available data and that is logically and internally consistent. When experienced anatomists see an unusual structure, they may be able to recognize instantly that it is new to science or is at least a significant variation. For physiologists, it is not so easy. If an experiment on photosynthesis does not come out as expected, it may be that a new type of photosynthesis is being discovered. It may be that the current theories of photosynthesis are not completely accurate and have made an erroneous prediction, or it may be that an experimental error has occurred. It can be difficult for students to appreciate the tremendous amount of careful, ingenious work that must be done just to establish that a particular theoretical metabolic pathway truly represents the reactions that occur in certain plants. Whereas it is relatively easy to determine that natural selection has resulted in the evolution of many types of leaves, stems, roots, flowers, xylem, phloem, and so on, it is much more difficult in physiology. Numerous differences in microhabitats, water availability, heat, cold, soils, pests, and plant diseases have resulted in diverse types of structures that are selectively advantageous under various conditions. It is logical to expect the same to be true of metabolism. We do know that there are several varieties of photosynthesis and respiration, and there may be others that have not yet been discovered. As you study the material in this section, keep in mind that, just as is true for structure, organization is of fundamental importance for metabolism. The chemical and physical reactions that constitute plant metabolism are highly ordered and not at all random. This orderliness is maintained by the input of energy acting on materials brought into plants from the environment. There are many types of order, and the information necessary to establish the proper reactions acting on the proper material is stored in the genes, both in the nucleus and in the plastids and mitochondria. The mechanisms by which the genes control the interaction of energy and matter, such that a plant of a particular species results, are discussed in chapters 14 and 15. Chapter 10. Energy Metabolism, Photosynthesis. Chapter opener image. The high-energy electrons moving through these power lines are dangerous and could easily kill a person. Photosynthesis also involves the movement of high-energy electrons, which, if not controlled properly, can and do damage chloroplasts and even the whole plant. There was no electricity flowing through these lines when they were being constructed, but a plant's photosynthetic pigments become energized as soon as they are synthesized, and they are even dangerous in autumn while chloroplasts are being dismantled. Whenever light strikes chlorophyll, an essential but risky process begins. Outline. Concepts. Energy and reducing power. Energy carriers. Reducing power. Other electron carriers. Photosynthesis. The light-dependent reactions. The stroma reactions. Anabolic metabolism. Environmental and internal factors. Light. Leaf structure. Water. C metabolism. Crassylation acid metabolism. Box 10-1 Plants and people, photosynthesis, global warming, and global climate change. Box 10-2 Alternatives, photosynthesis in bacteria and cyanobacteria. Learning objectives. After reading this chapter, students will be able to recall the method of energy transportation from energized pigments into endergonic reactions. Explain why plants need reducing power. List 4 important qualities of carbon dioxide and water as photosynthesis compounds. Describe the ideal photosynthetic pigment. Compare chlorophyll A to this ideal. Summarize the function of accessory pigments. Describe the ways in which the light-dependent reactions and the stroma reactions function together. Explain the ways in which photosynthesis affects Earth's climate. Did you know? The photosynthetic pigment chlorophyll absorbs mostly red and blue but is poor at absorbing green light. Green light bounces off and thus leaves appear to be green. Almost all energy used by animals, fungi, and everything else is the energy of sunlight captured by photosynthesis in plants, algae, and cyanobacteria. Light intensity must be just right. In habitats where sunlight is too intense, plants often have hairs or spines that shade the plant, if light is too dim. Not enough energy is available to allow life. Concepts. Probably the most important concept concerning cells and all of life itself is that living organisms are highly ordered, highly structured systems. The universe as a whole is constantly becoming less orderly, its disorder is increasing. Prokaryotes, protists, fungi, plants, and animals, however, represent phenomena in which particles become more orderly. A plant absorbs diffusely scattered molecules of carbon dioxide, water, and minerals and organizes them into organic molecules, cells, tissues, and organs. Each plant carries this out with such precision that each species of plant is easily distinguishable from others. After death, decay is the process by which an organism's molecules become more disordered and scattered, their entropy increases. Because living organisms are part of the natural world described by the laws of physics and chemistry, the decrease in the entropy of living organisms must obey physical laws. This is accomplished by putting energy into the living system, the source of energy being sunlight. To be accurate, we must consider the sun and life together. Atomic reactions that generate sunlight cause greater disorder in the sun than sunlight causes order in living organisms. The entire system becomes more disordered. Because there is no means of putting energy into an organism's body after death, an increase in entropy cannot be prevented. Sunlight maintains and increases the orderliness of life by two methods: directly, in the process of photosynthesis, which produces complex organic compounds, and indirectly, in the respiration of those organic compounds either by the organism itself or by another organism that eats it. These two methods of supplying energy and maintaining orderliness, photosynthesis and respiration, are the basis for a major, fundamental distinction in the types of organisms. Photoautotrophs are organisms that gather energy directly from light and use it to assimilate small inorganic molecules into their own tissues. Photoautotrophs include all green plants, all cyanobacteria, and the few bacteria capable of photosynthesis. Heterotrophs are organisms that cannot do this but instead take inorganic molecules and respire them, obtaining the energy available in them. Heterotrophs include all animals, all completely parasitic plants, all fungi, and non-photosynthetic prokaryotes. Gathering energy by taking inorganic material has the advantage that part of the material can be used as construction material instead of fuel. At least some of the amino acids, fatty acids, and sugars in food can be built into the organism's own polymers and the rest respired for energy. Photoautotrophs must build all of their own molecules using just carbon dioxide, water, and various nitrates, sulfates, and other minerals. Tremendously important consequences follow from the fact that photoautotrophs and heterotrophs differ in their sources of energy and building material. Sunlight and carbon dioxide do not need to be stalked, hunted, and captured. Thus, sensory organs, muscles, and central nervous systems like those of animals are unnecessary. Conversely, the ocean is full of microscopic bits of food, and animals such as sponges and corals can gather it the way plants gather carbon dioxide. The mode of nutrition has had overriding influence on the bodies and metabolisms of plants and animals. Tissues and organs are also either photoautotrophic or heterotrophic. Chlorophyllous leaves and stems are photoautotrophic, whereas roots, wood, and flowers are heterotrophic and survive on carbohydrates imported through phloem. During winter, if all leaves have abscised, the entire plant may be composed of heterotrophic tissues, and it maintains its metabolism by respiring stored starch. Figure 10. All organisms must obtain the energy needed to drive their metabolism. Most plants obtain energy through photosynthesis using the green pigment chlorophyll. All animals and fungi obtain their energy by either eating plants or by eating animals that ate plants. Some plants, called holoparasites, like this orange dodder, do not carry out photosynthesis. Instead, they send specialized roots called haustoria into the bodies of ordinary green, photosynthetic plants and extract sugars, water, and minerals from them and convert the sugars into the organic compounds they need. Close-up showing haustoria penetrating the leaf of a host. Table 10. Differences between photoautotrophs and heterotrophs. Tissues often change their type of metabolism. Young seedlings are white and heterotrophic while germinating underground. They survive on nutrients stored in cotyledons or endosperm. Seedlings become photoautotrophic only after they emerge into sunlight. Immature fruits may be green and photosynthetic. However, in the last stages of maturation, chloroplasts are converted to chromoplasts, and metabolism then depends on imported or stored nutrients. Chromoplasts are plastids that contain large amounts of pigments other than chlorophyll. Young leaf primordia are green, but they grow more rapidly than their own photosynthesis would permit. They have a mixed metabolism of photosynthesis and carbohydrate import. Photosynthesis is a complex process by which carbon dioxide is converted to carbohydrate. This involves endergonic reactions driven by adenosine triphosphate and requiring new bonding orbitals filled by electrons carried to the reaction by reduced nicotinamide adenine dinucleotide phosphate. Before this can happen, ATP and NADPH themselves must be formed in highly endergonic reactions driven by light energy. In order to understand this, you must first understand the nature of light and pigments along with the concept of reducing power. Energy and reducing power. Energy carriers. Energy enters the biological world through photosynthesis, a process that converts light energy to chemical energy. The sun's light is captured by certain plant pigments that use the energy in chemical reactions. Unfortunately, the energized pigments can enter into only 2 chemical reactions, although plants have thousands of different reactions in their metabolism. Several theoretical ways exist of transporting energy from energized pigments into endergonic reactions. Figure 10. The tail of chlorophyll contains only hydrogen and methyl functional groups, so it is hydrophobic and dissolves into the chloroplast's membrane lipids, immobilizing it. The porphyrin ring system of alternating double and single bonds acts as an antenna that captures light energy. The magnesium atom carries the electrons involved in photosynthesis. If the colored methyl group were an aldehyde group, the pigment would be chlorophyll B. 1. Allow the pigments to enter into every reaction necessary. A problem is that the energized pigments are large molecules, therefore, they are not very mobile and never move across membranes. Furthermore, they are too energetic. They can react with almost anything and would be difficult to control. Table 10. 2 methods of synthesizing ATP. 2. Allow the energized pigments to make one or several smaller, less energetic intermediates that can be moved and controlled easily. Such a method has evolved. Photosynthetic reactions produce ATP, an extraordinary molecule. Its high-energy phosphate bonds carry enough energy to force almost any reaction to proceed, and it can enter into almost every reaction for which energy is needed. In those that it does not enter, other energy carriers, often relatives of ATP, are involved. The most frequent is guanosine triphosphate, which also carries high-energy phosphate bonds. Although ATP is an essential molecule, it constitutes only a tiny fraction of the plant body. Each molecule is recycled and reused repeatedly, thousands of times per second. ATP is converted to ADP and phosphate by metabolic reactions, but the phosphate can be reattached with a high-energy bond by the reactions of either photosynthesis or respiration. Each molecule is an energy carrier, shuttling between reactions that release energy and those that consume it. There are 3 methods by which adenosine diphosphate can be phosphorylated to ATP. The first, photophosphorylation, involves light energy in photosynthesis. Animals, fungi, and non-chlorophyllous plant tissues cannot perform photophosphorylation because they lack the necessary pigments and organelles. Instead, they respire some of the high-energy compounds they have consumed as food or imported by phloem. Compounds with high-energy phosphate groups are produced, and these compounds force their phosphate onto ADP, making ATP. This is substrate-level phosphorylation. In the last stages of respiration, ADP is phosphorylated to ATP by oxidative phosphorylation. Each process occurs in a distinct site within the cell, and each captures energy from distinct types of exergonic reactions. Photophosphorylation occurs only in chloroplast sunlight, but substrate level and oxidative phosphorylation occur in all parts of the plant at all times. Reducing power. Earth's atmosphere is about 21% oxygen, therefore, many compounds are found in their oxidized form: carbon as carbon dioxide, sulfur as sulfate, nitrogen as nitrate, and so on. Oxidized means that an atom does not carry as many electrons as it could. In carbon dioxide, each oxygen can be considered to have pulled 2 electrons almost completely away from the carbon, and the carbon is said to be at a +4 oxidation state. This is speaking figuratively. Electrons spend more time near the oxygen than they do near the carbon, sulfur, or nitrogen, but they are not torn completely away. These bonds are covalent, not ionic. When electrons are added to an atom, it becomes reduced. Think of a reduction reaction as one that reduces the positive charge on an atom and an oxidation reaction as one that increases the positive charge. A preliminary rule of thumb is that oxidized compounds often contain a great deal of oxygen, whereas reduced compounds contain hydrogen. Oxygen has a strong tendency to pull electrons away from an atom and raise that atom's partial positive charge, but hydrogen becomes more stable by giving up electrons, reducing its partner's partial positive charge. Electrons can be transferred only between atoms or molecules, so each reaction in Table 10.4 is only a half reaction. Every oxidation occurs simultaneously with a reduction. The full reaction is known as an oxidation-reduction reaction, or redox reaction. Table 10.3 Calculation of oxidation states. Table 10.4 Reductions and oxidations. Whereas compounds in the environment are predominantly in the oxidized state because of our oxygen-rich atmosphere, most compounds in organisms are in the reduced state. Carbon is often in the form of carbohydrate, where its oxidation state is +0, nitrogen is frequently present as an amino group, NH, and sulfur is present as SH. Thus, in addition to needing energy, organisms also need reducing power, the ability to force electrons onto compounds. Reducing power is especially important to plants because they take in carbon dioxide and water, the most highly oxidized forms of carbon and hydrogen, and then convert them to carbohydrates, fats, and other compounds that are very reduced. Heterotrophs have less of a problem. When they consume plants, they get compounds that have already been reduced. They do need some reducing power, however, when they synthesize compounds that are very reduced, such as fatty acids. Just as with energy, an optimum solution for moving and handling reducing power, electrons, is to use small molecules that are semi-stable and mobile. The two molecules used most often are nicotinamide adenine dinucleotide and the closely related nicotinamide adenine dinucleotide phosphate. Both can pick up a pair of electrons and a proton, thereby becoming reduced to NADH and NADPH. When they reduce a compound by transferring their electrons to it, the proton is released and NAD or NADP is regenerated. Rather than having a large number of carrier molecules, the cell recycles each molecule, using it thousands of times a second as it shuttles between electron-producing and electron-consuming reactions. Because NAD and NADP take electrons away from other molecules, they are oxidizing agents, they oxidize the material they react with. It is also possible to say that the material has reduced the NAD or NADP. During the process, NADH and NADPH, two strong reducing agents, are formed. They have a powerful tendency to place electrons onto other molecules, reducing those molecules and becoming oxidized themselves. The tendency to accept or donate electrons varies greatly and is known as a molecule's redox potential. Cells contain a variety of electron carriers that differ in their tendency to accept or donate electrons. Figure 10.3 here, only the portion of NAD involved in carrying electrons is shown. The oxidized state carries a partial positive charge on the nitrogen and 3 double bonds in the ring. When it is reduced by 2 electrons, the positive charge disappears from the nitrogen, numerous bonding orbitals within the ring are changed, and the top carbon picks up a proton. No bonding orbitals are formed between NAD and the electron donor, therefore, NADH is free to diffuse away after picking up electrons. Similarly, as it donates electrons to some substrate, the ring bonding orbitals revert to the NAD form, none of which binds it to the substrate, so the NAD diffuses away. The same is true for NADP. In the upper reaction, 2 electrons are passed from a hypothetical molecule AH to NADP. AH has been oxidized to A, and NADP has been reduced to NADPH plus H. The NADPH is free to diffuse to another site, where it passes the 2 electrons onto another hypothetical molecule, B, reducing it to BH and becoming oxidized back to NADP. Table 10, 5 redox potentials of electron carriers. Compounds with a large negative redox potential tend to donate electrons and exist in the oxidized state, X, ferredoxin, NADP, FAD. Compounds with a large positive redox potential tend to accept electrons and exist in the reduced form, Cita+2, HO. Other electron carriers. Cytochromes. Cytochromes are small proteins that contain a cofactor, heme, which holds an iron atom. The iron carries electrons and cycles between the +2 and +3 oxidation states. Cytochromes are intrinsic membrane proteins. They are an integral part of the chloroplast's thylakoid membranes and cannot be removed without destroying the membrane. Consequently, they carry electrons only between sites that are extremely close together within a membrane rather than diffusing throughout the stroma as NADPH does. Plastoquinones. Plastoquinones, like cytochromes, transport electrons over short distances within a membrane. After they pick up 2 electrons, they also bind 2 protons. Their long hydrocarbon tail causes them to be hydrophobic, so they dissolve easily into the lipid component of chloroplast membranes. Plastocyanin. Like cytochromes, plastocyanin is a small protein that carries electrons on a metal atom, in this case copper. When oxidized, the copper ion is in the +2 oxidation state, but as it picks up the electron, it is reduced 1 level to the +1 oxidation state. Plastocyanin is loosely associated with chloroplast membranes. It can move a short distance along the surface, but it does not travel far. Figure 10, for in cytochromes, iron is not bonded directly to any amino acid but is held by heme, a box-like porphyrin ring similar to the portion of chlorophyll that holds the magnesium ion. The distinguishing feature of the quinone class of electron carriers is that each has 2 ketone groups whose carbon atoms are part of a ring structure. Photosynthesis. As the name implies, photosynthesis is a process that uses light energy to synthesize something. The term is so general that it could be applied to many different types of reactions, but whenever a botanist uses the term photosynthesis, the reaction being discussed is the combination of carbon dioxide with water to form carbohydrate, figure 10. Think about why these particular compounds are part of photosynthesis. First, both carbon dioxide and water are abundant and cheap, occurring almost everywhere in large quantities. The exception is the lack of water in severe deserts like the Sahara, where very little life exists simply because water is scarce. It is important to have a metabolism based on abundant compounds. It is necessary also for raw materials to be cheap, that is, the plant must be able to obtain them without expending much energy. Water and carbon dioxide are excellent because they diffuse into the plants automatically from soil, air, or water. Another important quality of carbon dioxide and water is that they are very stable and contain little chemical energy, so it is possible to deposit a large amount of energy into them. The carbohydrates they form are a good means of storing energy because all reactions leading to carbohydrate breakdown have high energy of activation barriers. Despite being energy-rich, carbohydrates are stable and chemically unreactive. Finally, both the reactants and the products of photosynthesis are non-toxic. It is safe to absorb large quantities of carbon dioxide and water and to store high concentrations of carbohydrates. Many substances that are critical for life are extraordinarily toxic if they become even slightly concentrated. Chlorine, sodium, ammonium, and many vitamins are just a few examples. Figure 10.5 Although this chemical equation succinctly summarizes photosynthesis, it reveals virtually nothing of the reaction mechanism or the many carriers and enzymes that participate. We cannot draw a reaction diagram because photosynthesis does not occur by the direct interaction of 6 molecules of carbon dioxide with 6 of water. However, the relative potential energies can be shown, indicating that this is an endergonic process. During photosynthesis, the carbon of carbon dioxide is reduced and energy is supplied to it, converting it to carbohydrate. The carbon atom in carbon dioxide is at the +4 oxidation state, whereas carbon atoms in carbohydrate are, in general, at +0. 4 electrons must be found and placed into new bonding orbitals around the carbon atom to reduce it. This is not easy because carbon is more stable in the oxidized state than in the reduced state. Carbohydrates such as wood and sugar can burn, releasing energy, but carbon dioxide does not. Therefore, a source of electrons and a source of energy are necessary for photosynthesis. The electron source is water, and the energy source is light. Water and light, however, do not act on carbon dioxide directly. Instead, they create the intermediates ADP and NADPH by a process called the light-dependent reactions. In a separate set of reactions, the stroma reactions, ATP and NADPH interact with carbon dioxide and actually produce carbohydrate. Figure 10, 6 light-dependent reactions of photosynthesis occur by means of membrane-bound carriers, but the actual formation of carbohydrate occurs in the chloroplast liquid. ATP-ADP and NADP-NADP diffuse between the two regions. No region of the chloroplast is far from a membrane, so the distances traveled are only a few hundred times the diameter of a molecule. Figure 10.7 on the left end of this electromagnetic spectrum are gamma rays. If treated as waves, they have extremely short wavelengths, less than 1 nanometer. If treated as particles, each quantum is highly energetic. Moving to the right, wavelengths become longer and quanta less energetic. The region to which our eyes respond, the visible region, is enlarged. These apples have an abundance of pigments that absorb all colors except red. Earlier, while still developing, they had the pigment chlorophyll, which absorbs red and blue strongly, but reflects green. The light-dependent reactions, the nature of light. Light is one small segment of the electromagnetic radiation spectrum, which encompasses gamma rays, X-rays, ultraviolet light, infrared light, microwaves, and radio waves, in addition to visible light. Radiation can be thought of and treated physically either as a set of particles called quanta, also called photons, or as a set of waves. The various types of radiation differ from each other only in their wavelengths and the amounts of energy each individual quantum contains. Short wavelengths have relatively large amounts of energy in each quantum, whereas long wavelengths have relatively little. Because humans use visible light for vision, we are more sensitive to and familiar with this region of the spectrum. We cannot see other wavelengths, but we can feel infrared radiation as heat, and our bodies react to ultraviolet light by becoming tanned or burned. We distinguish differences in quantum energy of visible light as differences in color. Most of us see all wavelengths from red through orange, yellow, green, blue, indigo, to violet. Certain insects see some near ultraviolet, but in general, all animals see in the range from 350 to 760 nanometers, which is also the radiation that plants use for photosynthesis. The nature of pigments. Most materials absorb certain wavelengths more than other wavelengths. If a substance absorbs all wavelengths except red, then red light either bounces off or passes through it, and the substance appears red to us. Any material that absorbs certain wavelengths specifically and therefore has distinctive color is a pigment, but we more often think of pigments as substances that absorb light as part of their biological function. Some pigments, such as melanin, absorb light and thereby protect other light-sensitive substances. Other pigments, such as those in flowers, fruits, or the skins of animals, are important for the light they do not absorb, which gives the pigments their color, allowing them to be useful to the organisms in attracting mates, pollinators, or frugivores or in hiding from predators. Photosynthetic pigments transfer absorbed light energy to electrons that then enter chemical reactions. Because only absorbed energy can be used, a theoretically ideal photosynthetic pigment would be black. It would absorb and use all light, not letting any escape. The pigment should at least absorb high-energy radiation instead of the fairly weak visible light. Rather surprisingly, the critical pigment, chlorophyll A, is not like this at all. It absorbs only some red and some blue light, letting most of the rest pass through, especially high-energy radiation. In general, plants only capture about 5% of the energy available in the electromagnetic radiation that strikes them. Why does chlorophyll lack what seem like ideal characteristics? Plants and people. Box 10-to-1 photosynthesis, global warming, and global climate change. Our atmosphere is critically important to life on Earth. Ironically, its composition is the product of that very life. The free oxygen we breathe is produced solely by oxygenic photosynthesis. There is no other source. Oxygenic photosynthesis originated 2.8 billion years ago. We know this because for millions of years the newly produced oxygen reacted with iron, forming a worldwide stratum of rust in ancient rocks. After all iron had been oxidized, free oxygen began accumulating in the atmosphere, and its concentration has been increasing ever since. Simultaneously, photosynthesis pulled carbon dioxide out of the atmosphere, converting it first to 3-phosphoglyceraldehyde and then to all of the other organic compounds that exist. Every single organic molecule started out as carbon dioxide snared by Ruby P carboxylase. Most organic molecules are digested and respired by aerobic organisms, a process that returns carbon dioxide back to the atmosphere rather quickly. But millions of tons of trees have died and fallen into stagnant swamps where a lack of oxygen prevented decay. The carbon in their wood, all of the cellulose, hemicellulose, and lignin, was converted to coal and did not return to the atmosphere. Petroleum also is probably derived from photosynthetically fixed carbon dioxide. The point is that respiration does not release all carbon back to the atmosphere, therefore, photosynthesis is gradually causing carbon dioxide concentration in the air to decrease. 3 groups of organisms have had especially important impacts on atmospheric carbon dioxide: coccoliths, mollusks, and humans. Coccoliths are microscopic algae that build shells of calcium carbonate, as do mollusks. When they die, their shells and the carbon they contain sink to the bottom of the ocean and decompose only slowly. All limestone and vast carbonate deposits on the ocean floor represent millions of tons of carbon dioxide removed from the atmosphere by clams, barnacles, and unimaginable numbers of algae. We humans were no different from any other aerobic organism until we made a fateful discovery: how to use fire. Since then, we not only oxidize food in our mitochondria, but we also oxidize wood, coal, oil, and gas, putting carbon dioxide back into the atmosphere and raising its concentration measurably. Why does the concentration of atmospheric carbon dioxide matter? Think of carbon dioxide as a pigment. Its absorption spectrum is low for visible light but high for infrared wavelengths. Visible light from the sun passes easily through the atmosphere. It is not absorbed by nitrogen, oxygen, or carbon dioxide. As it strikes Earth's surface, some is reflected immediately back out into space, and a small amount is absorbed by biological pigments such as chlorophyll in leaves or rhodopsin in eyes, where it powers photosynthesis or vision. However, most visible light has no effect other than to warm rocks, soil, and water, causing them to radiate the extra energy away as long wavelength. Infrared light. Many of these infrared quanta pass directly back through the atmosphere without hitting a carbon dioxide molecule because the concentration of carbon dioxide is so low, but many quanta are absorbed by atmospheric carbon dioxide molecules, causing them to become warmer. This energy is trapped in the Earth atmosphere system and warms our world. This is called the greenhouse effect because the glass in greenhouses works the same way, as does the glass in a parked car. Carbon dioxide is a greenhouse gas. An important balance exists between the atmospheric concentration of carbon dioxide and life. With less carbon dioxide, more heat would be lost and Earth would be frozen, like Mars. With more heat would be trapped and our world would be as hot as Venus, at 800 degrees Celsius, with lakes of molten lead. During the industrial age, we have been adding carbon dioxide to the atmosphere by burning oil, gas, and coal, and we have destroyed forest trees that can remove the carbon dioxide by photosynthesis. The concentration of carbon dioxide is increasing in the atmosphere, and the average temperature is also increasing. This is global warming, and it could cause mean temperatures to be 2 degrees Celsius or 3 degrees Celsius warmer in the next century. Global warming is having numerous consequences. First, surface water of the oceans is becoming warmer, therefore, more water evaporates into the air. Much of our weather in North America comes as winds blow eastward across the North Pacific. The water is cold and the air picks up only enough moisture to keep the Pacific Northwest wet. By the time it moves to the Central Plains states, it has so little moisture left that only grasses, not forests, thrive. But as surface waters of the Pacific become slightly warmer, vastly more moisture will evaporate into the wind and be carried to the Mississippi drainage basin. This increased rainfall could cause much better farming conditions in the Central Plains and catastrophic flooding in most river valleys where cities are located. El Niño years show the gigantic flooding that results from slight warming in just one area of an ocean. Global warming is also causing rapid melting of snow and glaciers in mountains and of ice caps in the Arctic and Antarctic. It is difficult to comprehend, but Antarctica is a large continent covered by ice 1 to 2 miles thick. As the world's ice fields melt, that water is added to oceans, increasing their volume and causing sea level to rise. Sea levels are rising even more because as ocean water warms, it expands. Coastal cities will be flooded, and so will coastal wetlands where ducks, geese, and hundreds of other species live. The additional freshwater flowing from melting ice caps and flooding rivers has other impacts. Freshwater is lighter, more buoyant than seawater. It spreads outward from a river's mouth as a cap, only gradually mixing with seawater as waves agitate the two. With more flooding, this cap becomes more extensive. Being fresh, marine algae cannot live in this layer, so they are forced into lower, darker waters where they cannot photosynthesize so well. The mixing of freshwater with seawater dilutes the nutrients in seawater, also slowing the growth of algae, and algae are of course the basis of the entire food web in the oceans. All ocean life is ultimately dependent on algae. Another consequence of global warming is that not all areas are being affected equally. Wind patterns are being changed such that certain areas are becoming warmer, others cooler, some wetter, and others drier. This is global climate change. If circulation of ocean currents like the Gulf Stream is altered, climates in vast areas of the world will change dramatically. At what point will we decide that weather patterns have truly changed and that a particular city should be abandoned because of frequent flooding? Some agricultural areas will become too dry to farm, causing at least personal misery as crops fail, perhaps causing massive starvation. New areas will become optimal for farming, but it will take a great deal of confidence to be certain that the new weather patterns are stable enough for people to risk starting again in an unknown area. Millions of lives will be disrupted, some suddenly and catastrophically in floods and others slowly and inexorably as conditions decline. In addition to the problems global climate change causes us, we are also forcing it onto all of the other organisms with which we share the planet and which bear no responsibility for the damage we are doing. Figure B10, 1A. After this tree is killed by the chainsaw, it will stop photosynthesizing and will stop removing carbon dioxide from the atmosphere. Even if converted to lumber and used as part of a house, it will eventually decay or rot or be burned, being oxidized back to carbon dioxide and increasing the amount of CO in the air. The Kyoto Protocol is a treaty designed to reduce production of greenhouse gases. Signed by 166 countries, it went into effect in 1994, but the United States, the largest producer of carbon dioxide, did not sign the treaty. Some people contend that if we do not act decisively, greenhouse gases will build up rapidly. Others point out that China and India, with combined populations of almost 2 billion people, are modernizing so rapidly that soon they will be producing more carbon dioxide than the United States is. They conclude that greenhouse gases will be produced as long as coal and oil are available and people need energy. One human interaction with the atmosphere seems to be going well. The atmosphere protects us from harmful radiation from space by absorbing high-energy cosmic rays, X-rays, and ultraviolet light. Very little of these wavelengths reach ground level. If not for water in the atmosphere, the first two would kill us outright. Ultraviolet light can induce mutations in our DNA and cause skin cancer and blindness, but atmospheric oxygen weakly blocks some ultraviolet light. Fortunately, a byproduct of its absorbing high-energy quanta is the conversion of oxygen to ozone, 3O. 2O, which is even more effective at blocking ultraviolet light. Man-made chlorofluorocarbons from air conditioners and cans of hairspray and deodorant, among other products, had been escaping into the atmosphere and destroying ozone, thus increasing the amount of ultraviolet radiation reaching us. Effort has been made to eliminate production of chlorofluorocarbons worldwide, and its levels in the atmosphere are dropping, thus allowing the ozone to accumulate again and continue protecting us. This one success story shows that we have the power to protect the environment that protects us. Figure B10, 1b. Tropical rainforests are not the only forests being damaged. Clear-cutting like this is common in the western United States and Canada. First, chlorophyll, like all other biological pigments, does not use high-energy quanta because they have too much energy. Each is so powerful that it would knock electrons completely away from the pigment, disrupting bonding orbitals and causing the molecule to break apart. Notice in Figure 10.2 that all bonds in the chlorophyll ring system are double bonds that alternate with single bonds. This bond system is excellent for absorbing quanta, but if even a single electron is knocked out, the entire structure becomes useless. It is selectively disadvantageous for a plant to produce a photosynthetic pigment that is destroyed by the light it absorbs. The molecule would break down, and all of the ATP that had been expended earlier in its construction would be wasted. Fortunately for our own molecules, the atmosphere's ozone layer protects us by absorbing high-energy radiation. Long-wavelength radiations, such as infrared and microwaves, have so little energy per quantum that they cannot appreciably boost an electron's energy. Instead, they make the pigment molecule warmer, as in a microwave oven, but this is not especially useful for chemical synthesis. Visible light contains just the right amount of energy per quantum. When one of these quanta is absorbed by the pigment, an electron is activated, raised to an orbital of a higher energy level. We say that the electron and the molecule go from the ground state to an excited state. Under the right conditions, this high-energy, excited electron can be used in chemical reactions. If it is not used, it returns to its original, stable ground orbital by emitting a new quantum of light, one with less energy and a longer wavelength than the one that it absorbed. The release of light by a pigment is called fluorescence. Figure 10.8 If a quantum has the wrong wavelength for a pigment, it passes through the pigment's bonding orbitals without being absorbed. Chlorophyll looks green because most green light passes through it. If the quantum has the correct wavelength, the correct amount of energy, it is absorbed, and the electron must move to a new orbital whose energy level corresponds to the electron's new energy load. The excited state is unstable. It may stabilize itself by having the electron emit enough energy to drop back to its original ground state energy level. The electron can also be stabilized by moving to a more stable orbital on an entirely different atom. This is the critical process in photosynthesis. Without this step, life would not exist. Figure 10.9 The absorption spectra of chlorophyll A and chlorophyll B and the action spectrum of photosynthesis. On the bottom axis is the wavelength of light, with short wavelengths to the left and long ones to the right. The vertical axis of the absorption spectra is the amount of light absorbed by the pigment. For the action spectrum, it is the amount of photosynthesis carried out. Chlorophylls absorb little of the very short wavelength light at 400 nanometers, and little photosynthesis occurs. However, light at slightly longer wavelengths, about 425 nanometers, is absorbed well by chlorophyll A, and photosynthesis proceeds. Quanta with intermediate wavelengths pass right through the pigment, and photosynthesis is low, but in the 650 to 680 nanometers range, considerable absorption occurs. Because the absorption spectra of chlorophyll A and B differ, more wavelengths are harvested. If the two matched perfectly, chlorophyll B would be useless. Two of the most useful pieces of information about a photochemical process are its action spectrum and the absorption spectrum of its pigment. An absorption spectrum is a graph that shows which wavelengths are most strongly absorbed by a pigment, whereas an action spectrum shows which wavelengths are most effective at powering a photochemical process. To initiate a photochemical process, light must first be absorbed. Therefore, the action spectrum of a process must match the absorption spectrum of the pigments responsible. The absorption spectrum of chlorophyll shows that it absorbs red light and blue light very well and other wavelengths only slightly. It would be better if it could absorb a greater number of wavelengths, but it simply does not. Chlorophyll is the essential photosynthetic pigment in all plants, algae, and cyanobacteria and has existed unaltered by evolution for about 3 billion years. To put this in perspective, the entire Milky Way galaxy takes 250 million years to make one rotation about its center, so in 12 full rotations of our galaxy, no alteration in the structure of chlorophyll has been selectively advantageous. Accessory pigments are molecules that strongly absorb wavelengths not absorbed by chlorophyll A. The absorbed energy is passed on to chlorophyll A. In effect, the accessory pigments overcome the narrow absorption of chlorophyll A and broaden the action spectrum of photosynthesis. We know that accessory pigments are involved because the action spectrum of photosynthesis does not perfectly match the absorption spectrum of chlorophyll A. The most common accessory pigments in land plants are chlorophyll B and the carotenoids. Algae have other types. Chlorophyll A and chlorophyll B are large, flat molecules with almost identical porphyrin ring structures. Their phytol tails are hydrophobic and dissolve into the lipid portion of the thylakoid membrane. When packed tightly in a membrane by their phytol tails, their porphyrin rings lie parallel to each other, which causes the electron orbitals of one molecule to interact with those of the two adjacent molecules. Hundreds of chlorophylls act somewhat like one molecule, and the energy absorbed by one can be rapidly transferred to another in a different part of the complex. This transfer, called resonance, allows chlorophyll B to absorb wavelengths that chlorophyll A would miss and then to transfer the energy to chlorophyll A for use in chemical reactions. Carotenoids are poor at this type of resonance and transfer only approximately 10% of their energy. They seem to be more important in absorbing excessive light and thus protecting chlorophylls. Rather than occurring at random, the pigments are held in place by proteins called light-harvesting complex proteins that are also embedded in the thylakoid membrane. For example, one well-studied protein is known to bind 14 molecules of chlorophyll A and B and as well as 4 carotenoids. The entire set of 300 chlorophylls, carotenoids, and their proteins are called an antenna complex. When light strikes any pigment of an antenna complex, the energy is transferred to a reaction center, a structure that contains a pair of special molecules of chlorophyll A whose properties differ from those of all other molecules of chlorophyll in the complex. The reaction center's pair of chlorophyll A molecules are the only ones that can actually donate an excited electron onto an electron acceptor molecule. Figure 10. There are two types of carotenoids. Carotenes lack oxygen, but xanthophylls have it. Both are accessory pigments that protect the chlorophyll from excess sunlight. Carotenoids are always present in leaves, but usually the abundant chlorophyll masks their presence. In autumn, chlorophyll breaks down, and we can see the carotenoids. Figure 10:11 Only special chlorophyll molecules, reaction centers, undergo the initial photochemical reaction of photosynthesis, but they are surrounded by other chlorophyll molecules as well as accessory pigments. Regardless of which pigment absorbs light, the energy is transferred to the reaction center. It seems strange for chlorophyll molecules to pass energy from one to another, but it is necessary because chlorophyll is so poor at absorbing light. Even in bright sunlight, any particular chlorophyll molecule absorbs only a few quanta each second. This is much too slow to keep the later steps of photosynthesis operating properly. By having an antenna complex, the several hundred pigment molecules all transfer their energy into one reaction center, which thus acts as if it is absorbing thousands of quanta each second. Also, this organization allows all the electron carriers and other crucial molecules to be clustered around just the reaction center chlorophyll a dimer rather than being scattered among all the photosynthetic pigments. When light energy reaches a reaction center in chlorophyll A, the electron is so unstable that it either reacts with almost anything or fluoresces, wasting its energy. Chlorophyll's electron must react only with the proper molecule, which should be close enough for the reaction to take place instantly, before fluorescence can occur. All pigments and carriers that work together are packed into a granule called the photosynthetic unit, and the thylakoid membranes are filled with millions of these granular arrays. Each photosynthetic unit contains an antenna complex and reaction center, but in some photosynthetic units, chlorophyll B is plentiful, and in others, it is less abundant. Those with little chlorophyll B have been named photosystem I. Those in which chlorophyll B is present at levels almost equal to are photosystem II. The photosystem I units are involved in the following reactions. Figure 10:12 Reaction center of photosystem I. A and B are two major proteins that bind chlorophyll. Many other components are clustered around these proteins. PC equals plastocyanin, FDX equals ferredoxin, F, and F are electron carriers. Photosystem I. The pair of chlorophylls of the photosystem I reaction center is given the special name P700 because they absorb red light of 700 nanometers most efficiently. The energy excites an electron of P700, which is then absorbed by a membrane-bound electron acceptor known as F. This is a transfer of an electron. No bonding orbital is formed. The exact chemical nature of F is not known, but it contains iron and sulfur and is sometimes designated Fe-S. When X absorbs an electron from P700, it becomes a powerful reducing agent, with a redox potential of -0.73 volts. The transferred electron is still extremely unstable, and the reduced F immediately passes it on to ferredoxin, which is also located in the thylakoid membrane. Ferredoxin is a small protein with an active site consisting of 2 iron atoms bound to 2 sulfur atoms. Reduced ferredoxin is also a strong reducing agent, with a redox potential of -0.43 volts. Electrons are passed from ferredoxin to an enzyme, ferredoxin NADP reductase, which then reduces NADP, converting it to NADPH, as its name indicates. Ferredoxin carries only 1 electron, but 2 are needed simultaneously to reduce NADP. Ferredoxin NADP reductase carries 2 electrons, but it can be reduced 1 electron at a time, then it transfers those 2 electrons together to NADP. Although NADPH is also a strong reducing agent, it is stable enough to move away from the membrane safely without the risk of reducing things indiscriminately, as the previous electron carriers might. Figure 10:13 In photosystem I, energy is absorbed by a pair of P700 chlorophyll molecules, raising 2 electrons to an excited energy level. From here, they pass on to Fe-S, then on to ferredoxin, and finally onto ferredoxin NADP reductase. After 2 electrons have reduced ferredoxin NADP reductase, they are transferred simultaneously to NADP, reducing it to NADPH plus H. Figure 10:14 The two photosystems work together to transfer electrons from water to NADPH. The scale on the left indicates reducing power, the redox potential. The higher a molecule is in the chart, the greater its capacity to force electrons onto another molecule. Because of its shape, this diagram is called a Z-scheme. Photosystem 2. Photosystem I efficiently produces NADPH, but the reaction center P700 chlorophyll loses electrons during the process. In this oxidized state, bonding orbitals could easily rearrange, causing the molecule to break down and be destroyed. There must be a mechanism that adds electrons back to the P700, reducing it so that it can work repeatedly. The mechanism that reduces P700 is photosystem 2. Photosystem 2 can be best described by working backward from photosystem I. A molecule of plastocyanin, which contains copper, donates an electron to the chlorophyll of the photosystem I reaction center. The plastocyanin is now oxidized, lacking an electron. It must reacquire one because it also is too expensive a molecule to donate just one electron and then never work again. It receives its new electron from a complex of cytochrome molecules, called the cytochrome B6F complex, which in turn gets an electron from a molecule of plastoquinone. This receives electrons from another carrier, Q, a molecule of quinone, which in turn receives electrons from pheophytin. Pheophytin is actually a chlorophyll, a molecule that does not contain a magnesium atom. Pheophytin becomes oxidized as it donates an electron to Q, so it must obtain another electron, which it does when a chlorophyll, a molecule, absorbs light and is activated. This is a different chlorophyll from the one in photosystem I. It is the reaction center of photosystem 2 and has the name P680. We may seem to be going in circles, taking an electron from one chlorophyll A, P680, to pass it on to another chlorophyll A, P700, which then sends it to an ADP, but the physical differences between the two molecules of chlorophyll are crucial. The one in photosystem 2 gets new electrons from water, not plastocyanin. The important thing is that water is cheap enough to just throw away after the electrons are removed. Water breaks down into protons, which the plant uses, and oxygen, which it discards. Whereas all electron carriers are large, expensive molecules that the plant must construct itself, water is simply brought in. The electrons are stripped off, the protons are used, and the oxygen is discarded through stomata. 10 quanta are needed to break down each water molecule. Photosystems 1 and 2 together are an efficient system. Electrons are passed from water to P680 in photosystem 2, their energy is boosted by light, and then they move through an electron transport chain, the various electron carriers, to P700 in photosystem I. Their energy is boosted by light again, and they pass through a short second electron transport chain to an ADP, reducing it to NADPH. This last step requires that protons be added to an ADP. These protons are present in the water surrounding the membrane. It would be simpler if photosystem I could receive electrons directly from water, but that does not happen. Besides, the electron transport chain between P680 and P700 is necessary for the production of ATP. The synthesis of ATP. The light-dependent reactions produce the reducing agent NADPH that actually places electrons onto the carbon of carbon dioxide in the stromal reactions, but the stromal reactions are highly endergonic and must be driven by being coupled to the exergonic splitting of ATP. The necessary ATP is also generated by the light reactions, but the process is indirect. It is photophosphorylation because light is involved, but a more specific name is often used, chemiosmotic phosphorylation. To understand it, we must take a closer look at the structure of chloroplasts. The inner membrane of chloroplasts folds inward, forming flattened sacs called thylakoids. In certain regions, these swell slightly and form rounded vesicles. All thylakoids in one region form vesicles at the same spot, so they occur in sets called grana. Thylakoids that lie between grana are frets. The liquid surrounding the thylakoid system is the stroma, but notice especially that there is another compartment, the thylakoid lumen. Figure 10-15 grana are stacks of small thylakoid vesicles compressed together. Frets are regions of thylakoid that connect one granum to another. The lumen of the thylakoid region is continuous with that of the fret region. The liquid surrounding all of the thylakoids is stroma. The thylakoid lumen is a critically important compartment because some of the enzymes and electron carriers of the photosystems are embedded in the membrane layer facing the lumen, whereas other enzymes are in the membrane layer facing the stroma. Reactions that break down water and produce oxygen and protons are located on the lumen side of the thylakoid membrane, in the granum areas. This membrane is not permeable to protons, therefore, as light reactions run, the thylakoid interior accumulates protons, and their concentration increases. The molecules of ferredoxin NADP reductase that generate NADPH are located on the other side of the membrane, facing the stroma. The protons they attach to NADP are those present as a result of the natural breakdown of water, HOH plus OH. As protons are absorbed, their concentration in the stroma decreases. Furthermore, during electron transport between P680 and P700, the electron carrier plastoquinone moves a proton from the stroma to the thylakoid lumen every time it carries an electron between pheophytin and the cytochrome B6F complex. This also contributes to the increased concentration of protons in the thylakoid lumen and to the decreased concentration of protons in the stroma. Figure 10-16 The water splitting, proton-producing reactions of photosystem 2 take place on the lumen side of the thylakoid membrane. Plastoquinone is like an ADP in that when it picks up electrons it also picks up a proton. This occurs on the stroma side of the membrane, but the reduced plastoquinone must diffuse to the other side of the membrane to pass electrons onto the cytochrome Bf complex. The proton then dissociates and is deposited in the lumen, adding to the growing pool of protons. When NADPH is formed, it picks up protons from the stroma. This and the plastoquinone pumping result in a deficiency of protons in the stroma. Protons return to the stroma by passing through ATP synthetases. Their passage is exergonic and powers the phosphorylation of ADP to ATP. Figure 10-17 In non-cyclic electron transport, electrons flow through the Z-scheme from water to NADPH. Cyclic electron transport is much simpler. Electrons flow from P700 to plastoquinone, which carries a proton to the lumen and returns the electron to P700. The strong difference between the concentrations of protons inside the thylakoid lumen and exterior to it in the stroma quickly becomes so powerful that protons begin to flow out of the lumen through special channels in the membrane. These channels are complex sets of enzymes that can synthesize ATP from ADP and phosphate. The entire complex is called ATP synthetase. The ATP synthetase of chloroplasts is known specifically as the CF-CF complex. CF is the portion of the enzyme spanning the membrane where the actual proton channel is located. CF is the portion of the enzyme that phosphorylates ADP to ATP. The power required to force phosphate onto ADP and establish the high-energy bonding orbitals of ATP comes from the flow of protons through the ATP synthetase channels. In a car battery, electron flow through wires powers the starter. In chloroplasts, proton flow through ATP synthetase channels powers phosphorylation of ADP to ATP. When electrons flow smoothly from water to NADPH, the process is called non-cyclic electron transport. The chemiosmotic potential that builds up does not produce quite enough ATP for the stroma reactions. There is too little ATP relative to the amount of NADPH produced. This problem is overcome by an alternate route for electrons. After they reach ferredoxin in photosystem I, they can be transferred to the plastoquinones of photosystem II instead of being used to make NADPH. The plastoquinones carry the electrons along just as though they had gotten them from Q and use their energy to pump more protons into the thylakoid lumen. This is cyclic electron transport, and with it, chloroplasts make extra ATP without making extra NADPH, thus producing ATP and NADPH in the proper ratios for the stroma reactions. Cyclic electron transport is a simple light-powered proton pump, and similar types occur in bacteria. This may have been the original power system that evolved first, billions of years ago. Figure 10:18 in the yellow area are the first steps of the stromal reactions, also known as the C cycle. The product is two molecules of 3-phosphoglyceraldehyde. Some of this is transported out of the chloroplast, and the rest undergoes reactions that form a new molecule of the acceptor, RuBP. At various times, acids such as phosphoglycerate and malate are written as phosphoglyceric acid and malic acid. The IC acid ending refers to the whole acid. The 8 ending refers to the acid's anion, the negatively charged portion left after the proton dissociates. In protoplasm, most of the acids occur as free anions, not intact neutral acids still holding their protons. The stroma reactions. Conversion of carbon dioxide to carbohydrate occurs in the stroma reactions, also called the Calvin-Benson cycle or the C cycle. These reactions take place in the stroma, mediated by enzymes that are not bound to thylakoid membranes. In the first step, an acceptor molecule reacts with a molecule of carbon dioxide. Because RuBP contains 5 carbons and 1 more is added from carbon dioxide, you might expect a product that contains 6 carbons. However, the new molecule breaks apart immediately. While still on the enzyme, stable bonding orbitals cannot be formed between all 6 carbon atoms while so many oxygen atoms are present and pulling electrons to themselves. Instead, orbitals rearrange and 2 identical molecules are formed that each contains 3 carbons, 3-phosphoglycerate, hence the name C cycle. The abbreviation PGA is often used for 3-phosphoglycerate. The enzyme that carries out this reaction has many names. The most common is RuBP carboxylase. This is one of the largest and most complex enzymes known, a giant complex of 2 kinds of protein subunits. There are 8 copies of a small protein, each with a molecular weight of 14,000 to 15,000 daltons, and 8 copies of a large protein, each with a molecular weight of 53,000 to 55,000 daltons. The entire enzyme has a molecular weight of about 480,000 daltons. Not only is the tertiary structure of each protein subunit important, but their quaternary structure as a complex is critical. When all 8 subunits are properly assembled, the large subunits form the functional active site. RuBP carboxylase can constitute up to 30% of the protein in a leaf, making it the most abundant protein on Earth. Without it, there would be almost no life at all. All photosynthesis that produces oxygen is mediated by this enzyme. A few photosynthetic bacteria use an enzyme composed of just large subunits, lacking any small ones. RuBP carboxylase is crucial to the production of food. Without it, heterotrophs would starve. Like chlorophyll A, RuBP carboxylase is by no means ideal. Its active site recognizes and binds to carbon dioxide only poorly, and it has low substrate specificity, frequently putting oxygen rather than carbon dioxide onto RuBP. Yet this enzyme is highly conserved evolutionarily. The amino acid sequences of RuBP carboxylase from all plants are virtually identical. Apparently, all mutations that cause any change in structure, however slight, disturb the active sites and are selectively disadvantageous. It is important to realize that the first step of the stroma reactions is carboxylation only. Electrons and energy are added in the next 2 steps. ATP donates a high-energy phosphate group to the 3-phosphoglycerate, converting it to 1,3-diphosphoglycerate, which then is reduced by NADPH to 3-phosphoglyceraldehyde. A phosphate comes off in this step also. The carbon is now both reduced and energized. The rest of the stroma reactions are complex, but the important point is that as they operate, some 3-phosphoglyceraldehyde can be taken out of the chloroplast and used by the cell to build sugars, fats, amino acids, nucleic acids, basically anything the plant needs. The rest of the PGAL remains in the chloroplast and undergoes several more stroma reactions, which convert it to RuBP, the original acceptor molecule. The principle involved is important. To incorporate carbon dioxide, the plant needs the acceptor Rub-P, and the two react on a one-to-one basis. To assimilate large amounts of carbon dioxide, the plant either needs large amounts of Rub-P or needs to use a few Rub-P molecules repeatedly. Plants use the second strategy. As 3-phosphoglyceraldehyde is formed, some of it is reconverted to Rub-P by the rest of the stromal reactions, and some is exported to the cytoplasm. The chloroplast does not need to import quantities of Rub-P from the rest of the cell, it just recycles the small amount that it has. Imagine a chloroplast that has 1 million carbon atoms inside it as the various intermediates of the stromal reactions. After 3 carbon dioxides have been assimilated, there are 1,000,003 carbon atoms. When one molecule of 3-phosphoglyceraldehyde is exported, the carbon pool returns to 1 million, and a steady state is maintained. In very young leaves with growing chloroplasts, little or no 3-phosphoglyceraldehyde is exported, it is retained, and the pools of C metabolites increase in numbers of molecules. Anabolic metabolism. 3-phosphoglyceraldehyde is an amazingly versatile molecule. Using it plus water, nitrates, sulfates, and minerals, plants construct everything inside themselves. The entire fabric of the organism can be synthesized. This is also the basis of all animal metabolism because animals either eat plants or eat other animals that eat plants. Most biological molecules are larger than 3-phosphoglyceraldehyde, so it must be rearranged and altered in the cytoplasm to build up larger, more complex molecules. This constructive metabolism is called anabolism, and it consists of anabolic reactions. Anabolic pathways are numerous, but 2 are especially important with regard to energy metabolism: the synthetic pathways of polysaccharides and fats, which are storage forms of energy, and carbon. The NADPH and ATP produced by photosynthesis are excellent sources of energy, but they cannot be stored for even a short time. They are so reactive and unstable that they would break down. A plant cannot stockpile them to survive times when photosynthesis is impossible, nor can they be transported over long distances. Therefore, even if leaves had an abundant supply, roots would starve. Several types of storage compounds have evolved that solve these problems. 1. Short-term storage. ATP and NADPH can be used within the cell and last only briefly. 2. Intermediate-term storage. The simple sugar glucose and the disaccharide sucrose are stable enough to be moved from cell to cell, either in the vascular tissue of a plant or in a bloodstream. They are also sufficiently stable to last for weeks or months. A problem with storing large quantities of monosaccharide or disaccharide is that they cause cells to absorb water by osmosis. 3. Long-term storage. Starch is a large, high molecular weight polymer of glucose, too large to be transported. It is even more stable than glucose, lasts for years, and does not cause the cell to absorb water. Lipids are an even more concentrated storage form of energy that can be synthesized rapidly and stored in large quantities. The synthesis of polysaccharides. The anabolic synthesis of glucose is gluconeogenesis. In reactions similar to those of C metabolism, part of the PGL exported to the cytoplasm is converted to dihydroxyacetone phosphate. One molecule of this condenses with one molecule of unconverted 3-phosphoglyceraldehyde to form the sugar fructose 1,6-bisphosphate. This loses a phosphate to become fructose 6-phosphate, and part of this is rearranged, converting it to glucose 6-phosphate. Both fructose 6-phosphate and glucose 6-phosphate are versatile, useful molecules that enter many metabolic pathways. In plants, the glucose 6-phosphate is polymerized into polysaccharides, amylose, amylopectin, or cellulose. Both amylose and amylopectin are components of starch. Amylose is an unbranched polymer of glucose, containing between 500 and 20,000 glucose residues. Amylopectin is also a polymer of glucose, but it branches and can be extraordinarily large, with as many as 1 million glucose residues in each molecule. The enzymes that add glucose to amylose and amylopectin can only work at the ends of the molecules. Because amylose has only one end, it is synthesized slowly, whereas amylopectin might have thousands of enzymes simultaneously adding glucose to the thousands of its branches. Similarly, amylopectin can be digested back to glucose monomers much more rapidly than amylose. The speed of starch degradation is important to us animals as well as to plants. Most of our food is rich in starch, and its digestion begins even before we swallow because our saliva contains salivary amylase. We digest the amylopectin portion of starch rapidly, which makes glucose available to our bloodstream quickly. In contrast, the amylose portion of starches is digested more slowly. Plants store starch in both chloroplasts and amyloplasts. Other than a few rare exceptions, animals never synthesize starch. We store glucose as glycogen instead. During daylight hours, as photosynthesis is occurring, most of the 3-phosphoglyceraldehyde is retained within the chloroplast and gluconeogenesis converts it to glucose. It is polymerized to starch within the chloroplast, and by afternoon, chloroplasts are usually swollen because of the large starch grains they contain. At night, the starch is depolymerized back to glucose, which is transported from the chloroplasts to the cytosol. Much of it is then converted to sucrose, loaded into phloem, and transported to various parts of the plant that need carbohydrate. By morning, most chloroplasts have little or no starch left in them. In organs involved in long-term storage of starch, such as potato tubers and starchy seeds like wheat and rice, sugar moves from the phloem into parenchyma cells, then enters amyloplasts where it is repolymerized to starch again. Figure 10:19 Gluconeogenesis is an anabolic pathway in which large molecules are built up from small ones. This process may occur in chloroplasts, amyloplasts, or cytosol. Most of the sugar we eat and drink comes from sugar cane, which is a type of grass with large stems more than 2 m tall. This harvester cuts the shoots off at ground level, slices the stems into pieces called joints, and loads them into the cart behind the tractor. Pieces of leaves are so light they blow away. These joints of sugar cane must be taken to a refinery and have their sap squeezed out and refined into raw sugar within 18 hours. Immediately after the stems are cut in the field, enzymes begin to convert the disaccharide sucrose into the monosaccharides glucose and fructose, both of which are less valuable than sucrose. Sucrose is obtained by boiling cane sap to concentrate the sucrose until it becomes so concentrated it crystallizes. The crystals are centrifuged out and stored in giant piles as raw sugar, shown here. The remaining liquid is molasses. Raw sugar is converted to refined sugar by dissolving it again and reboiling it to recrystallize it, after which it is filtered. The raw sugar in this warehouse will last indefinitely if it is kept dry. Environmental and internal factors. A plant's photosynthesis is affected by its environment in many ways. Light. From a plant's viewpoint, light has 3 important properties: quality, quantity, and duration. Quality of sunlight refers to the colors or wavelengths it contains. Sunlight is pure white because it contains the entire visible spectrum. During sunset and sunrise, sunlight passes tangentially through the atmosphere, and a large percentage of the blue light is deflected upward. Consequently, light at ground level is enriched in red, which is easily visible. This period of red-enriched light lasts only a few minutes and probably has little effect on photosynthesis. At noon, sunlight passes nearly vertically through the atmosphere, more blue light is transmitted, and even though the blueness of the sky suggests that all reds, greens, and yellows have been blocked, in fact, enough of all of these wavelengths penetrate to Earth's surface to allow efficient photosynthesis. This is true of plants in deserts, grasslands, and the top layer, the canopy, of a forest. However, herbs and shrubs that grow near soil level in a forest are understory plants, and the light that they receive has already passed through the leaves of the canopy. As light penetrates those leaves, red and blue are absorbed by chlorophyll, so the dim light received by understory plants is especially depleted in these critical wavelengths. It is selectively advantageous for them to have extra amounts of accessory pigments so that they can gather the wavelengths available and pass the energy on to chlorophyll A. Similarly, algae that grow near the surface of lakes or oceans receive complete light, but water absorbs red and violet. Algae at deep regions receive mostly green and blue light and must have special accessory pigments capable of absorbing these wavelengths sufficiently. Quantity of light, which refers to light intensity or brightness, is affected by several factors. More light is available for photosynthesis on a clear than on a cloudy day. Understory plants receive dim light, lower branches and branches on the shaded side of a plant receive less light. Plants growing in the shadow of mountains or in deep canyons receive much less light than plants that grow on slopes that face the sun. Plants growing near the equator receive intense light because the sun is always more or less directly overhead at noon, whereas plants near the poles receive very little light. Even during the summer the sun is low at noon, and light is scattered by the atmosphere. Figure 10:20 Plants growing in the shade of other plants receive not only dim light, but light depleted of red and blue. Not all leaves of a tree receive equal amounts of light. This self-shading is more severe in latitudes farther away from the equator. Colors indicate the percentage of full sunlight that reaches the various parts of a tree. Figure 10:21 Many kelps grow in deep ocean water and receive mostly green and blue light. Their accessory pigments absorb these wavelengths, therefore, the kelps appear yellow-brown to us. Think about how intensity of sunlight varies during the day and affects photosynthesis. Examine the solid line labeled 300 ppm county in Figure 10:22. This was derived from many experiments in which the rate of photosynthesis was measured for plants grown under different intensities of light, but all with 300 ppm carbon dioxide in the air. Near the left side at point, those plants that received dim light absorbed little carbon dioxide, whereas those grown in brighter light absorbed more carbon dioxide. Under these normal levels of carbon dioxide, light is the limiting factor. Photosynthesis is slow on dull, overcast days but faster on brighter days. At point in the graph, plants that received more light did not photosynthesize faster than those that received slightly less. Where the curve turns flat, there was enough light to saturate the process. In these conditions, the limitation was lack of carbon dioxide in the experiments in which more carbon dioxide was available. Photosynthesis went faster. Thus, at point, carbon dioxide was the rate-limiting factor. Under conditions of adequate water, light, and fertilizers, the rate of photosynthesis in whole plants increases with increasing carbon dioxide concentrations, being 30% faster with concentrations between 600 to 750 ppm. It should be noted that the experiment in Figure 10:22 was performed in the 1970s, but by 2015, atmospheric carbon dioxide already reached 400 ppm. At point, light was so intense that it damaged the plant by overheating it and bleaching the pigments. At point, if the lack of light prevents photosynthesis from proceeding faster, there must be adequate amounts of carbon dioxide. As soon as ATP and NADPH are produced, they move to the stroma and are used by the waiting enzymes and carbon dioxide, then ADP and NADP diffuse back to the thylakoid membranes and wait for another quantum. Conversely, at point, where light is bright and the low concentration of carbon dioxide is rate limiting, there is so much light that as quickly as ADP and NADP come to the thylakoids, they are reprocessed immediately into ATP and NADPH. These then move to the stroma, where they must wait for a carbon dioxide molecule. Figure 10:22 Light and photosynthesis, details are given in text. The unit ppm is parts per million. For 300 ppm, in 1 million liters of air, 300 liters are carbon dioxide. These grapevines have abundant sunlight, water, and fertilizer. They could probably photosynthesize more rapidly only if more carbon dioxide were present. Figure 10.23. While young, the leaves of Dusty Miller are completely obscured by trichomes, protecting the leaf from strong sunlight and insects. These cacti live in environments where sunlight is extremely intense. Their spines are so abundant and closely spaced that they shade the stem and prevent chlorophyll from being damaged. Plants of Dudleya bretonii are protected from excess sunlight and water loss by a thick layer of white wax. This particular wax strongly reflects ultraviolet light. To the left of the light compensation point, it appears that there was no photosynthesis, even though some dim light was provided. The problem actually lies with the technology of measuring photosynthesis. Either the amount of carbon dioxide absorbed or the amount of oxygen released must be measured, but both of these gases are involved in respiration as well as photosynthesis. To the left of the light compensation point, photosynthesis was absorbing carbon dioxide more slowly than respiration was releasing it. Thus, it appears as though no photosynthesis occurred. The same problem arises when we try to measure respiration. Photosynthesis distorts the measurement, but then we can turn off the lights and stop photosynthesis. The light compensation point is the level of light at which photosynthesis matches respiration. Plants that are grown for a long time in conditions below the light compensation point respire faster than they photosynthesize. They gradually consume their reserve carbohydrates and fats and starve to death. For plants grown in light brighter than the light compensation point, photosynthesis exceeds respiration, and the extra sugar can be used for growth and reproduction. When considering the effects of the amount of light, we need to consider the shape and organization of a plant. Photosynthesis in most ordinary leaves is saturated even on overcast days. 1/4 to 1/2 of ordinary sunshine is all most leaves can use. In contrast, many lower leaves on trees and bushes are shaded by upper leaves, and they may be exposed only to small flecks of sunlight that move irregularly over the leaf surface. Even on sunny days, many of the lower leaves are not receiving enough light to photosynthesize at their maximum capacity. Consequently, whereas an upper leaf may be saturated in partial sunlight, most plants are not. As light intensity increases, more of their lower leaves photosynthesize faster, so total photosynthesis for the entire plant increases. In the brightest environments, the air is so clear that sunlight is frequently too intense during summer months. Protective adaptations are necessary, and in many species, mechanisms have evolved that provide shade. A common method is the production of a thick layer of dead trichomes, plant hairs. A heavy coating of wax can also reflect light, and cutin is especially good at absorbing the more harmful short wavelengths. Part of the value of carotenoids and other accessory pigments is that they shade the chlorophyll, absorbing some of the most damaging wavelengths. The intensity of light, the actual number of quanta that strike a given area per unit time, is greatest at noon in midsummer when the sun is most directly overhead, and is less whenever the sun is lower, morning, evening, and winter. Light may be too intense at midday but optimal when the sun is lower. Some species have adapted to this by means of vertical leaves. The lamina face is exposed fully only in mornings and afternoons, but at noon, only the leaf edge is exposed. In other plants, leaves orient vertically automatically when stressed. They wilt and hang down. Understory plants of forests are adapted to low light. If a roadway is cut into a forest and plants adjacent to the cut are suddenly exposed to full sunlight, the shock may cause them to wilt and die. The same phenomenon occurs when trees are blown down during storms, floods, or avalanches and the surrounding plants are exposed. The duration of sunlight refers to the number of hours per day that sunlight is available. At the equator, days are 12 hours long throughout the year. Farther north or south, days become longer in summer. Maximum length occurs near the poles, where the day is 24 hours long in midsummer, and only night occurs in midwinter. In middle latitudes, winter days are short, and sunlight is weak because the sun is so low in the sky. Under these conditions, even evergreen plants are unable to undergo very much photosynthesis. However, because temperatures are low, the plants are growing little and have a low rate of respiration. Even deciduous trees and biennials can survive by means of stored nutrients. During summer, days are longer and light is brighter because the sun is higher in the sky. The amount of energy obtained by photosynthesis easily exceeds that consumed by respiration and growth. In many plants, longer days cause greater amounts of photosynthesis, but in others, chloroplasts become so full of starch that photosynthesis stops even though light is present. At night, starch is converted to sugar, which is then transported out of chloroplasts and can be used for growth or stored in amyloplasts in tubers, corms, or other such organs. By morning, leaf chloroplasts can resume photosynthesis. Figure 10.24 These living stone plants of African deserts conserve water in several ways. They have only 2 leaves at a time. When 2 new ones form, the old 2 die. Not enough water is available for 4 leaves. The leaves are fleshy and pressed together, such that they form a cylinder with minimal surface area through which water can be lost. This is a plant of Haworthia cooperi. Its short stem is located several centimeters underground, and it produces many cylindrical leaves that are just long enough to reach the soil surface. The tips of the leaves, visible here, have transparent epidermis, and the mesophyll is also transparent because it has no intercellular spaces. Consequently, light enters through the window-like tip and then passes deep into the rest of the underground leaf, where cells with chloroplasts are located. Photosynthesis actually occurs underground. Because most of each leaf is subterranean, they stay cooler in summer, they are less visible to animals, and the air in the soil is richer in carbon dioxide than is air above ground. This transverse section of a leaf of aloe vera is so transparent you can read through it. It is excellent for transmitting light to parts of the leaf that are underground. Leaf structure. Leaf structure of most temperate and tropical plants is quite standard, palisade parenchyma above and spongy mesophyll below. This structure is excellent for absorbing carbon dioxide but inefficient for conserving water. If plants of hot, dry habitats had this leaf architecture, they would have to keep their stomata closed so much of the time that they would starve. Instead, their leaf cells are frequently packed together without intercellular spaces. Water loss is reduced because the small internal surface area retards water evaporation, but with so little surface area, it is difficult to dissolve carbon dioxide from the air into the cytoplasm. This slows photosynthesis, but apparently this trade-off, slow growth versus water conservation, is selectively advantageous. Another method of minimizing water loss while maintaining photosynthesis is to reduce external surface by means of cylindrical leaves. Water movement from interior air spaces to exterior air is minimized because so few stomata are present. Photosynthesis is reduced because absorption of carbon dioxide is slowed. Water. The amount of water available greatly affects photosynthesis. Most plants keep their stomata open during the day, permitting entry of carbon dioxide, but water is inevitably lost. At night, carbon dioxide cannot be used, and stomata are closed, retaining water within the plant. If the soil becomes dry and water is not readily available, a plant keeps its stomata closed even during the day, and carbon dioxide cannot enter. The small amount of carbon dioxide produced by the respiration of the leaves can be reused photosynthetically, but this is a minor amount. In addition to numerous structural modifications that conserve water, metabolic adaptations also exist. 2 of the most important are C metabolism and crassylation acid metabolism. Figure 10.25 Light intensity exceeded the light compensation point for these trees just after 6 a.m. and photosynthesis increased rapidly. However, by early afternoon, photosynthesis dropped even though light and temperature were adequate. The problem was a lack of water, and probably stomata had begun to close around noon. On a cloudy day, water stress never became a problem and stomata remained open. Even though there was less total light than in, there was more photosynthesis for the day. C metabolism. An important factor for plants is the amount of water lost for each molecule of carbon dioxide absorbed. This ratio is called the plant's water use efficiency. Ideally, this ratio is low. Carbon dioxide diffuses into a leaf faster if its concentration in air is higher or if its concentration inside leaf protoplasm is lower. Plants can do nothing about the carbon dioxide level in the external air, and many also have poor control over the protoplasmic concentration. Ruby P. carboxylase has a low affinity for carbon dioxide. As carbon dioxide concentration drops, enzyme substrate binding slows. Even while carbon dioxide is still rather abundant in protoplasm, the enzyme is only rarely picking it up. With this relatively high concentration, carbon dioxide diffusion into the leaf is slow, whereas water loss may be high. Under normal conditions, each molecule of Ruby P. carboxylase fixes only 1 to 12 carbon dioxide molecules per second. This is extremely slow. Most enzymes catalyze thousands of reactions per second. Ruby P. carboxylase occasionally binds to oxygen instead of carbon dioxide, acting as an oxygenase and producing one molecule of 3-phosphoglycerate and one of phosphoglycolate. This latter molecule is transported from the chloroplast to peroxisomes and mitochondria, where some of it is converted to the useful amino acids glycine and serine, but much of the phosphoglycolate is broken down to 2 molecules of carbon dioxide.